keeping our squids healthy and clean. I also do my best to keep the lab clean and in tip-top shape.
Can you be a little louder, Rachelle? I'm sorry.
Sure. Sometimes I assist my fellow interns and grad students with their projects in the lab. This presentation is divided up into two major sections. In section 1, I will talk about what I do to help the squid, and in section 2, I will talk about my lab work. Section 1, all about squid. The Hawaiian bobtail squid is known for its short size and short lifespan, and it's suitable as a model organism. The squid has a mantle that contains the light organ. This light organ contains the heart, the stomach, and the gills. The squids have 8 arms and 2 water jets. The squids have 8 arms and 2 longer feeding tentacles, which all contain suckers. These suckers can be used to grab and hold on to food. The squids have 2 large eyes, and their eyes are very similar to the structure of human eyes. The squids use their water jets as a means of rapid movement, both away from predators and towards prey. Their siphon is located on the mantle cavity and expels water and waste. Squids breathe using gills. They move by squirting water from the mantle through the siphon, using a type of movement called jet propulsion. This experiment involves a symbiotic relationship between the Hawaiian bobtail squid and the bacteria called Vibrio fischeri. The squid's immune cell determines which bacteria to accept and which to destroy. And unlike many symbiosis, these squids maintain a relationship with only one specific bacteria called Vibrio fischeri. The symbiotic relationship between the squid and its bacteria can be compared to a flashlight and its battery. The squids have a built-in flashlight located in its mantle where the light organ is. They use the battery, the Vibrio fischeri acts as a battery, and the light organ is called the cilia, and it takes in the Vibrio fischeri from its surroundings, and from there the bacteria set up a colony. The squids can glow once this Vibrio fischeri colony reaches its critical mass. We call this glow bioluminescence. This demonstrates a symbiosis relationship because the bacteria colonize the squid, and the squid, as a result, glow due to the bacteria's colonization. The squids are able to find the Vibrio fischeri bacteria on Earth. We want to see if they can find this bacteria in space. We found that under microgravity conditions, the bacteria were able to successfully colonize the whole squid. This means that microgravity does not affect the squid-bacteria relationship, which proves that this process can happen in space. Okay, the squids are kept in a growth chamber. We also call this growth chamber the squid chamber. They live in salt water, and their optimal temperature is 23 degrees Celsius. Every week I check the ammonia, nitrate, and nitrite levels of the squid. It's important that we keep them at a certain pH. The lower the pH, the better, because high pHs can kill them, basically. Water salinity. I use what we call a refractometer to measure the salt concentration in the water. The optimum salt concentration is 45%. And on the right side is the percentage, and on the left side is what we use to measure specific gravity. So when I first enter the squid chamber, I check the water levels. Then I check the clutches. By doing this, I open the squid tank and I take out a clutch and I flip it over to see if there are any eggs. If there are any eggs, I move them into a separate tank. If there are no eggs, I put them back in the squid tank, and yeah, that's it. Step 3, I feed the squid shrimp. Each squid requires 3 shrimp. And step 4, I change the water in each of the clutch tanks. Step 5, I record data. The data that I record is the number of clutches that hatch and the number of squids that hatch from those clutches. In step 6, I do this once a week. I clean tanks, check the pH levels and the water salinity. So in this picture, we can see 3 clutches. They're different clutches, and as the day 20 approaches, they turn black, and this is because the squids are approaching their hatching date. We see a picture here of clutches in squid tanks. These are the tanks that I keep the clutches in until their hatching date. Before their day 20, I move them to bowls, and then when they hatch, they look like little, like probably 1-2 millimeters in length, baby squids. Okay, section 2, lab work. Okay, so autoclaved, sorry. Okay, RNases are found in all cell types and organisms, from prokaryotes and eukaryotes. These enzymes generally have high specific activity, meaning tiny amounts of contamination in an RNA sample is sufficient to destroy the RNA. We use 2 methods of sterilization to get rid of these RNases, autoclaving and baking. These are some examples of lab materials that we autoclave and bake. The boxes on the top right contain tip tubes. On the bottom left, we have a mortar and a pestle wrapped in tin foil. On the top left, we have a picture of a spatula. And on the bottom right, we have a beaker that contains a 0.5 milliliter test tube. Okay, bead beating tubes. I spent most of my time in the lab on bead beating tubes.
We feel bad for you. I'm sorry. This is not my job.
Bead beating tubes are used to release RNA and DNA from cells. On the right-hand side, we have different sizes of beads. We combine them into one, like we measure specific amounts and combine them into one whole, and we put them in a test tube. Then we place our sample in the test tube and then vortex it, so that way the beads can rip the cell apart and outpours the DNA and RNA. RNA extraction. I also spent my time in the lab with Diana and Kimberly doing RNA extraction. We sequence, RNA sequencing is basically sequencing of a transcriptome. A transcriptome is the full range of messenger RNA or mRNA molecules expressed by an organism. We use RNA to study how microbial mats and calcium carbonate evolved. Having a basic understanding of how microbial mats work make it easier to understand how life evolved on Earth. On the left, we have our method that we use to carry out RNA extraction. So I want to give a special thanks to Space Florida, Tony Gannon, the Foster Lab, including Dr. Jamie Foster, Giorgio, Diana, Joanny, Lexi, Mary, Kim, the Sugar Lab, the Nicholson Lab, and everybody else that helped to contribute. I also have a video of the squid eating a shrimp. So
You might use a pointer and tell them where the shrimp is on the bottom.
So the shrimp,
These guys.
so the squid, I can fast forward, I'll rewind that.
Rachelle, is that the slow motion video?
Yeah.
It happens much faster.
Yeah, not for the shrimp.
We can let it load, and you could probably take some questions.
Yes.
What are your future plans?
I want to go to, well, first I have to go to undergrad.
Yeah. It'll be quick. It will go fast.
I have to go to I want to go to med school eventually.
We're working on that. We're working on grad school.
Yes, Joanne? Oh.
Oh.
Yes, sometimes those squiddies will ink and reject some ink if they're disturbed. Does the ink interfere with their behavior such that you can't feed them for a few days, or they're skittish, or they in some way alter their behavior?
No, they don't. They just they eject ink when they feel threatened or after mating, but it doesn't interfere with anything. Any more questions? Yes.
Shrimp does it eat in a day?
Does it what?
How many shrimp does it eat?
Oh, each squid gets eats 3 shrimp. Yes.
Do you have like a main female squid that produces the egg, or multiple female squids that produce the eggs?
We have, I think, 8 squids in all. Probably more, but we just mate them, and all of them produce eggs.
So
There's not a main one.
is it like 4 females, 4 males, or 1 female and 7 males?
Well, there's 2 males, and there's a lot of more than 8 females.
Alright. Do they have names?
I can name them.
Try not to get too attached to them. But we can name the guy Fernando. We have one.
Yes.
Any more questions? Yes?
What do you think was the biggest thing you took away from this summer internship? What was your favorite thing that you learned?
The favorite thing that I learned was the RNA extractions, and I also liked the squid. I learned a lot from that. I like grinding mets. With the
It's probably calming.
yeah.
Because you get to use liquid nitrogen,
Yeah.
right? That's what the liquid nitrogen.
That's always cool.
Thank you, Rachelle.
I don't think I can see it in this anyway.
You know, there's females.
Okay, ready?
Okay.
1, 2, 3.